जरा मैसेज करें कि कौन कौन से लोग आ चुके हैं, लाइव अटैच हो चुके हैं। अगर थोड़े से अभी अवेलेबिलिटी नहीं लग रहा है, of course we are associating after a long gap. लाइव कौन कौन आ चुके हैं जरा मैसेज करें आप लोग। We are just associating here to continue the next part of lipids and proteins. We are discussing today on the catabolic reactions or processes related with the lipids and proteins. In last interaction, we discussed on the catabolic pathways associated with the carbohydrate or glucose metabolism. And in that class, we we suggested you that the protein catabolic reactions of proteins or the catabolic reactions of the lipids both are associated with either glycolysis or the Krebs cycle pathway. In that class, I told you that the fatty acids are catabolized at second stage and produced the acetyl coenzyme A. And that acetyl coenzyme A is entered in the Krebs cycle. Likely, I also told you that the different amino acids entered in the different stages of Krebs cycles, either at the acetyl coenzyme level or alpha glutaric acid level, or at the succinyl coenzyme level, or as the oxaloacetic acid level. That means the catabolic reactions, if you consider the catabolic reactions as a whole in any biological system or at the cellular level, that means the Krebs cycle is associated with all these three catabolic reactions. Either the energy is produced from the carbohydrate, or the energy is produced from the proteins, or the energy produced is produced from the lipids. And the Krebs cycle is actually the junction of all these three and particularly the acetyl coenzyme A level of the acetyl coenzyme A. And in last class also, again I am repeating here the brief of the catabolic reactions. The protein is at first lysed by the proteolytic enzymes to provide to release the peptones. Then the peptones is catalyzed by the proteolytic enzymes and produce the amino acids. It is well known, and we will discuss later on in classes, that the enzyme is substrate specific. We all know the enzyme is substrate specific, but if we but it is better to say that the enzyme is bond specific. Of course, it is the substrate specific. It is true that the enzyme is substrate specific, but it is more accurate to say that the enzyme is bond specific, because we observe the structure organization of the protein, then we say we observe that the nature of the peptide bonds is different with respect to the due to the differentiation in the nature of the amino acids. So the number of the proteolytic enzymes are more than the number of the enzymes associated with the lysis of the bond in the carbohydrates or in the lipids. So the proteolytic enzymes, the different proteolytic enzymes are responsible for the production of the amino acids, and then the amino acids is catabolized by the biochemical pathway and enters at the different levels of the Krebs cycle or the acetyl coenzyme A. Likely, we discussed very well the catabolic in the catabolic reactions of the carbohydrate. So I am not repeating here the steps associated with the carbohydrate metabolism, catabolism. Second, the lipid. The lipid, we know very well that mostly the lipids are present in the form of triglycerides. That is the glycerol associated with the 3 fatty acids. Then, if the lipid breaks enzymatically, then it produces glycerol and fatty acids. Of course, if we study the lipid is considered as the primary reserve energy, primary reserve substance to produce the energy. Lipid is considered, the fatty acids is considered as the primary source to conserve the energy, and it is estimated that it commonly produces 18% of the total energy. It commonly produces the 18% of the total energy of the living system. Of course, like the Krebs cycle and the glycolysis, the activation of the fatty acids. Of course, here again, the lipid is present in the cytoplasm. It is present in the cytoplasm of the cell. But the beta oxidation, that is the main steps associated with the catabolic reactions, that is referred as the beta oxidation, is observed in the mitochondria. The beta oxidation, why we say the beta oxidation, just I am going to discuss it later on. And the glycerol is catabolized or entered in the energy production system through the glyceraldehyde 3-phosphate of the glycolysis. That is, glycerol enters in the production, in the catabolic pathway through the steps of the glycolysis. It is not entered at the Krebs cycle level. In the glycolysis, and the glycerol is converted into the glyceraldehyde 3-phosphate. Then the glyceraldehyde 3-phosphate follows the same path of the glycolysis, which is followed by the glyceraldehyde 3-phosphate. Then it converts into the pyruvic acid and in acetyl coenzyme A. Then it enters in the Krebs cycle. The another component of the lipid, that is the fatty acid, is at first activated. That is what we say, that the activation of the fatty acid. And the fatty acid is activated by the association of the enzyme coenzyme A. Enzyme coenzyme A is at first associated with the carboxylic group of the fatty acid. We will know that the fatty acids are long chain carboxylic acid. Fatty acids are commonly long chain carboxylic acid, and it is commonly more abundantly present from the carbon 12 to carbon 16. And here, after the acylation, it is activated. Again, as we discussed in my previous online class, that at first you remember the steps associated with the any biochemical pathway. And here the steps are fatty acid activation. Activated fatty acid is converted into the acylase. Then acylase is converted into the enzyme coenzyme A. Enzyme coenzyme A is converted into the hydroxyacyl coenzyme A. Hydroxyacyl coenzyme A is converted into the ketothiolase, and the ketothiolase is converted into the two less carbon fatty acids plus coenzyme A. Then coenzyme A is released. That means the complete beta oxidation pathway requires 5 steps. So at first it is necessary to remember the steps. First step is the activation of the fatty acid to acylose. Second is the conversion of the acylose to the enzyme coenzyme A. Third is the conversion of enzyme coenzyme A to hydroxyacylase. Fourth is the conversion of the hydroxyacylase to the alpha ketothiolase. And then the fifth step is the production of the two less numbered fatty acid plus coenzyme A. That means the 5 steps are associated with the catabolic pathway of the fatty acids. Then you at first remember that all these 5 steps, and after the remembering of these 5 steps, you analyze the function or the chemical changes or the chemical reactions or the chemical steps. The first stage is the activation of the fatty acyl coenzyme A to form activated acylose. In this step, the coenzyme A is associated with the carboxylic group of the fatty acid. And in this process, when the coenzyme A is associated with the carboxylic group of the fatty acid, it requires the enzyme. And the enzyme is like the, as we observed in the enzymatic actions of the carbohydrate, the enzyme is very simple, the thiokinase. And here, the thiokinase, the step utilizes ATP. 1 unit of ATP is utilized in the activation of the fatty acid to form the acylose. And here, it is activated, and after the activation, acylose is second step is the conversion of the acylose to enzyme coenzyme A. It is the second step of the beta oxidation of the fatty acid. The second step is the conversion of acylose to enzyme coenzyme A. It is clear here that in the enzyme, that means presence of a double bond. Presence of a double bond. Here, the FAD linked dehydrogenase enzyme is associated. Flavin linked acid dehydrogenase enzyme is linked, and it is not completed in a single step, but at first the third hydrogen of the third carbon is activated and acts as a free radical to remove the two hydrogen from alpha and beta carbon. And as the alpha and hydrogen ions of the alpha and beta carbon group, that is the H plus from the alpha carbon and H plus from the beta carbon, a double bond is formed between the alpha and beta carbon. And now the released hydrogen is accepted by the FADH2, FAD, and it converts into the FADH2. And this product is the enzyme coenzyme A. Then the third step is the conversion of the enzyme coenzyme A to hydroxyacylase coenzyme A, hydroxyacyl coenzyme A. Here, in the hydroxyacyl coenzyme A, one water is used, one H2O is used here. And then H2O ionizing or the nuclear reactions, of course, is the not free radical, it is the nuclear reactions, and it is associated with the breaking of double bond, and again a ketonic component is developed, that is the keto group. Ketonic component or ketonic group is formed at the beta carbon. Then after the or in the nut cell, it is better to say that the association of the hydrogen to the enzyme coenzyme A is responsible for the breaking of the double bond, and thereafter the intra-reorganization of the molecules between the alpha and beta carbon. It is responsible for the formation of the keto group at the beta carbon. And now the conversion is responsible for the reorganization is responsible for the formation of beta ketothiolase. And here the hydroxyacyl coenzyme A to the beta ketothiolase is associated with the activity of NADH linked dehydrogenase enzyme. That means the hydroxyacylase coenzyme A is formed after the reorganization and breaking of the double bond in between the alpha and beta carbon. Alpha and beta carbon of the enoyl coenzyme A, and it forms the hydroxyacyl coenzyme A. Then the hydroxyacyl coenzyme A is responsible for the formation synthesis of a alpha ketothiolase by the activity of the NADH linked dehydrogenase enzyme. Now, when we consider the fifth step of the beta oxidation of fatty acid, then the alpha carbon and beta carbon in association with the coenzyme A is splitted and form the coenzyme A, acetyl coenzyme A, that is the acetyl coenzyme A plus fatty acid, which has two less carbon than the initial carbon, because these alpha and beta, that is two carbons, are removed from the parent fatty acids in the form of acetyl coenzyme A. Then the acetyl coenzyme A is entered in the Krebs cycle. So it is clear from here that each fatty acid is reduced in one step of the Krebs cycle by 2 carbon. And if any fatty acids has the 12 or 14 or 16 carbon chain, that means the fatty acid is repeatedly follows the steps, and in each step 2 carbon is reduced from the fatty acids. And now once the fatty acid is activated, it forms the if it has the 16 carbon, that means the 8 acetyl coenzyme A is formed, and then each acetyl coenzyme A enters in the Krebs cycle and produces the energy. That means the fatty acids is considered at the high energy components. It has the high energetics value. Then these fatty acids commonly it is stored, and it is responsible for the production of the energy, particularly during the fasting and starvation conditions. Now it has the energetic value, and in this way the fatty acids are catabolized and breaks into to produce the energy. Then we come on the catabolic activities of the protein. The proteinous commonly the catabolism of the protein for energy production is prohibited. Is prohibited. It is protein is commonly not involved in the production of the energy. But sometimes, if the extreme need of energy is required and where the mm, other possibility is not available in the living system, only in that condition protein is used for the production of energy within the living system. Commonly, it is not used. But if it is used, then the protein is dissociated into the amino acids, and amino acids take part in the production of the energy. And here, in my last discussion, uh, I told you that the some components of the some amino acids, roughly 10 amino acids are entered in the Krebs cycle through the acetyl coenzyme, and three, sorry, 5 amino acids are entered through the alpha ketogluteric acid. 3 amino acids are entered through the at the level of the succinyl coenzyme, and 2 amino acids are entered at the level of the fumaric 2-oxaloacetic acid level. But the catabolic reactions of the protein, the two significant processes which we are not discussed in the last class, because that class was particularly associated with the catabolism of the carbohydrate. That two processes are important processes are associated with the protein catabolic reactions. That is responsible for the conversion of the one amino acid to another amino acid, and transfer of the amino group from one biochemical molecules to the another biochemical molecules. And here, the one is the transamination and another is the deamination. That means the transamination and deamination both are significant biochemical processes associated with the protein catabolism. And as we observed from the name, that is transamination. Trans means the transfer of uh, amino group from one alpha carbon to another alpha carbon of the another amino acid. That is referred as the transamination. And generally, it is a process. If we want to remember it, then it is a process in which alpha amino group of amino acid is transferred is transferred to the alpha carbon atom of one of the 3 keto acids. Just I am saying that the transfer of the amino group to alpha amino group to alpha carbon of the another amino acid. That means if the alpha amino acid has own amino group, then there is no need of the transfer of the amino group. That means here the amino group of the alpha carbon is commonly transferred to the another alpha carbon, which alpha carbon of the amino acid, which has the keto functional group. That means, which is why so it is. I am saying that it is a process in which the alpha amino group of amino acid is transferred to the alpha carbon atom of one of the 3 keto amino acids, that is alpha ketogluteric acid, pyruvate, and oxaloacetic acid. These 3 acids has the keto functional group, that is pyruvic acid, alpha ketoglutarate, that is the alpha ketogluteric acid, and oxaloacetate, that is the oxaloacetic acid, which is present in the mitochondria and cytoplasm both. ये एमिनो एसिड्स माइटोकॉन्ड्रिया में भी हो सकते हैं और ये एमिनो एसिड्स साइटोप्लाज्म प्लाज्मा में भी हो सकते हैं. And here one coenzyme is required. The coenzyme is the pyridoxal phosphate. Pyridoxal phosphate. Here the pyridoxal phosphate is associated. Enzyme is it is very simple to say the name. The transaminase. The common enzyme because is the transamination. The enzyme associated with the transfer of the amino group, that is the transaminase. It is very simple, but it is the significant point that the coenzyme associated with the transamination is the pyridoxal phosphate. Pyridoxal phosphate. Why it is important? Because pyridoxal phosphate is the derivative of vitamin B6, and that is involved in the transfer of the that is involved in the enzymatic activities of the transaminase enzyme. The another significant process is the deamination. Deamination, simply the term itself is sufficient to explain the name. Deamination means the removal of the amino group. That is the deamination. Removal of the amino group is the deamination, and it is the process. Just if you want to say, then what is this? It is the process in which amino acid is catalytically, it is not self, but it is catalytically removed from alpha amino acids and convert into keto form. If the alpha amino acid is catalytically removed, and when the alpha amino acid is removed, then it converts into the keto group, that is the keto amino acids. The deamination process is either oxidative or hydrolytic. That is the again significant point for your examination, because commonly if we observe the biochemical pathway of the different catabolic pathway of the biochemical components, then if one step is occurred, then one step has the one nature, one biochemical nature. But here the deamination, and deamination is either occurred through the oxidase enzyme or through the dehydrogenase enzyme. That means it depends on the conditions, and it depends on the nature of the amino acids available for the deamination. That means here the process is occurred by the oxidative steps also and by the reductive steps also. Both oxidative and reductive steps are responsible for the deamination. And here these components are responsible for the conversion of the amino acids to nature of the amino acids, and that nature of the amino acids is responsible for the catabolic actions of the biochemical pathways. And all the significant parts we were discussed in my last class, and after the conversion of the amino acids, that amino acids is entered in the Krebs cycle or in the other components. The another part which is not associated with the catabolism of the catabolic reactions associated with the energy production, but is significant. That is the production of urea. Why? Because it is associated with the protein metabolism and it is also associated with your coming examinations. Then I think that it is important, it is more convenient to explain the urea cycle here. And just I am giving the information for the urea cycle. That urea cycle is also occurred in the mitochondria. And it is responsible for the production of urea. We well know that the ammonia is highly toxic components. Ammonia is highly toxic components, and in the more developed animal groups, the ammonia is converted into the less toxic components urea, and that urea is excreted through the uric acid. Through the urine, and that synthesis of urea and the biochemical pathway of the urea was at first explained by Krebs, but one association is here. Krebs and Hanslitt are responsible for the explanation of the urea cycle. And here, in the urea cycle, alpha amino group, it is associated again with, of course, it is the catabolic pathway, but it is not associated with the production of energy. In the catabolic pathway, of course, some part of the catabolic pathway, and if, of course, it is synthesis, it synthesizes the urea, then it requires some energy. It requires some energy because it is responsible for the synthesis of urea. And of course, the alpha amino group of the any amino acid is at first converted into glutamic acid. Glutamic acid या glutamate. Glutamine alpha amino acid is converted into glutamic acid or glutamine. And here the enzyme associated is the glutamine synthetase. That means the glutamine synthetase is responsible for the synthesis of glutamate and from alpha amino groups of the amino acids, and it is highly toxic. It is highly toxic, and in the next step, the glutamic acid or glutamine is deaminated. Process is the deaminated to aspartic acid in one way, and in one way carbonyl phosphate. That carbonyl phosphate is enters that carbonyl phosphate is enters in the urea cycle, and the carbonyl phosphate in association with the ornithine, the carbonyl phosphate in association with the ornithine for citrulline. I am repeating here. At first, the alpha amino acid converts into the glutamic acid. The glutamic acid is deaminated to form aspartic acid and carbonyl phosphate. The carbonyl phosphate in association with the ornithine forms citrulline. The step of conversion of carbonyl phosphate in association with the ornithine to citrulline requires 1 molecule of carbon dioxide. 1 molecule of carbon dioxide and 2 molecules of ATP. That means 2 ATP and 1 CO2 are consumed here. Then 1 carbon dioxide and 1 amino group enters in the urea cycle through ornithine and forms the citrulline. Again, the citrulline is converted into the argiosuccinic acid. Arginosuccinic acid. And here, in the conversion of the citrulline to arginosuccinic acid, another amino group deaminated to form the aspartic acid is entered. Then one amino group is entered. Well, alpha amino group is entered at the stage of the ornithine to citrulline, and another alpha amino group is entered through citrulline to arginosuccinic acid. That means if you observe the urea, that the NH2CO NH2, it is the chemical form of the ammonia, urea. And here, one carbon and one amino group enters at the ornithine to citrulline, and one another group, amino group, is entered at the citrulline to arginosuccinic acid. It is the complex structure, complex biochemical structure. That means the necessary component, two amino groups and one carbon, are entered in the form, in the urea cycle. One at the ornithine to citrulline level and one at the citrulline to arginosuccinic acid level. Then arginosuccinic acid is converted into arginine. And at this stage, fumaric acid is formed. That fumaric acid moves to the Krebs cycle to take the further action. Then the arginine is again converted into ornithine enzymatically. Upper steps are some biochemical intermediates, but here the enzymatically arginine is converted into the ornithine. And that ornithine, which receives one carbon and one alpha amino group at the ornithine to citrulline level and one amino acid to the citrulline to arginosuccinic acid level, release the 1 unit of urea. That means the both amino groups and CO2 received by the different steps from ornithine to arginine are released here. And again ornithine is formed, and now ornithine again repeats the cycle. And this cycle is referred as urea cycle. In this way, urea is repeatedly formed by the utilization of the ATP, 3 units of the ATP. And another step is also that is associated with the synthetic activities, that is the synthesis of the uric acid. And the synthesis of the uric acid is a complex process. Synthesis of the uric acid is a complex process in which released amino groups first forms the purine ring. In your syllabus, some anabolic components are also, anabolic reactions are also provided for in your syllabus, which we will discuss in later on. But in short, I am just saying that the alpha amino groups are for the pathway for the synthesis of the uric acid is complex process, but the alpha amino acids released are at first convert the purine ring, and then the purine ring is converted into the xanthine. The purine ring is converted into the xanthine, and finally the xanthine is oxidized into the uric acid. This is the pathway for the synthesis of the uric acid. In this way, we are just discussing on some fundamentals of the uh, catabolic reactions associated with the fatty acids and proteins. And you all are suggested that if you feel any problem, then either you give me message, because the uh, conversation, probably the conversation on the YouTube live is not possible. And so for each live class, you are free to give to ask any question if you have, by putting some message. And after today, I will provide always 5 to 10 minutes or 5 minutes for your message. I will wait for your message. And if you have, or any viewer have, because I suppose that all are my students. And if any, if viewers or anyone have any problem or any question, you put the question. और इफ़ यू हैव नो क्वेश्चन, जो जुड़े हुए हैं, अगर आपके पास क्वेश्चन नहीं है तो आप एक मैसेज डाल दीजिए, क्वेश्चन नहीं है। हम स्ट्रीमिंग को टुडे कंप्लीट करते हैं और देखते हैं कि वी आर कब हम लोग क्लास तक पहुँचे। एंड मोस्ट प्रोबेबली इन कमिंग फ्राइडे हम आपको ऑफलाइन क्लास में बुलाएंगे, क्योंकि ये जो भी क्वेश्चंस हैं, वी आर जस्ट talking on the theoretical aspects, but practical and applied aspects क्या क्वेश्चन हो सकते हैं, इस पर कुछ इलस्ट्रेशंस भी हो सकते हैं जो कि ऑनलाइन नहीं संभव है यहाँ पर। उसके लिए क्लास में हम आप लोग को बुलाएंगे और आप लोग को वहाँ पर बताया जाएगा। आई सपोज दैट कि आप लोग को कोई ये नहीं है, समस्याएं नहीं है। एंड नाउ थैंक्स टू ऑल, जो भी जुड़े हुए हैं उनको बहुत बहुत धन्यवाद, बहुत बहुत बधाई। और आप लोग जुड़े।